All right, the next chapter that we're going to look at is the description of carbohydrates, which I don't know why this book does this, but we've already talked about degradation of sugar, and now we're going to introduce what sugars are. So it's kind of a little bit backwards. I thought you might, probably need to know what sugars look like before we started actually utilizing them. But really, in this case, what we're really going to look at in detail, or a little bit more detail, is, okay, what kind of carbohydrates do we find? What kind of functions might they have in the body? And we're going to see that carbohydrates actually are more than just energy molecules, which is kind of what we've seen or kind of classified them in terms of how we have interacted with the sugars. So carbohydrates are used in the body in a lot of different places. Energy sources, degradation to produce ATP. Got that, done that, covered that. Can do the stoichiometry and tell you how many we can make. But they also form structural frameworks of the fundamental types of molecules that are in the body for function. So like DNA, RNA, ATP, FAD, electron carriers. A lot of things have sugars as the base, and we make derivatives of those sugars, and then that complex has a goal and works in the body to facilitate reactions, to store information and do things. A lot of times we don't actually look at the sugar of those. We just talk about its function, but if you go down to its key component, we have that sugar that is the bases. Also, we're going to find that they're going to be structural in cells and exoskeletons, and so not nutritional but structural, holding things together, holding shape, and actually can create very hard substances within the cell. And then the sugars are going to be informational, and so we are going to display sugars on the outside of cells. And the three-dimensional structure that the sugars make is individually bound by something else, and then that binding of recognition sends a information signal between two cells. And so we've got lots of things that sugars can do. The classes, we have simple sugars, and so monosaccharides, disaccharides, and very short linkages of sugars together. They are moved around in the bloodstream and they're utilized fairly quickly. We have polysaccharides, which we have long chains of sugars. Both of these can either be energy storage or can be structural in nature. And then we have glycoconjugates, and that means I have a sugar attached to something and they will do different things. And so in the glycoconjugates, these are kind of maybe a little bit more interesting in terms of function. Here we have a protein embedded in the membrane, and the sugar component is going to be informational. So it kind of tells what that cell is going to do. Here we've got lots of sugars that are bound. There's a little tiny protein in the middle that are holding all that, but this is going to be in the between cells, and it's going to allow the cells to be held together, just kind of like stickiness. It also allows space where there's a lot of water in which nutrients can flow through. So it can be cushioning and allows small molecules to move through. And then sugars for information can be attached not to just a protein, but also to a lipid, so that it's giving informational ideas. One interesting thing about sugars and cells, when the sugars are placed, exclusively, just about, on the outside of a cell. What you don't ever see is sugars that are inside the cell to the cytosol where the sugars are facing. It's always oriented outwards. Polysaccharides can be found in two forms. One called homopolysaccharides, the other is heteropolysaccharides. As you can see, the homopolysaccharides would be a polymer of a single sugar, and we can have the unbranched and branched. And then heteropolysaccharides are polymers that have repeating structures of sugars, and it can be branched or unbranched. Most of the time, the homopolysaccharides can be storage for energy. Some of the homopolysaccharides are structural. The heteropolysaccharides are mainly structural in nature, and we're going to find both in the biological systems. Glucose is stored as a polymer, and we have three different forms that we'll look at. Animals store glucose in the form of glycogen, and plants store glucose in the form of starch. There are two different versions or two different structures, amylose and amylopectin. The starch, when you take a look at amylose, is a very simple, unbranched polysaccharide. And what we see is we have a 1 to a 4, and it's in the alpha configuration. So that means this whole strand is below this sugar. So it follows and it comes down, and it will stair step down. We write the bond in a very unusual way so that we can get it linearly and save space in a book. What we really find in terms of the structure, when we put these together, remember, they are not this structure, but actually chair forms of the sugar. And we see that when it's below, it places the sugar below one ring and then the next sugar. And what happens in that particular structure is that it creates this curlicue effect when starch is packed. Starch is an easily accessible form of glucose that we can ingest, created by plants, and it is degraded by the enzyme amylase, which is wonderful. The structure amylose, the enzyme amylase breaking it down. It breaks it down into maltose, which is a disaccharide of sugar, of glucose, maltotriose, and dextrin is anything larger than 4, but smaller than the entire species. Maltase converts the first two into glucose, and then dextrinase can convert dextrin into glucose itself. And so this is a store of glucose, easily accessible. Our enzymes can break down this fairly quickly, and we can make sure that blood glucose levels are very high if we eat lots of starch. Amylopectin is another starch, and it is similar in that it is an alpha 1,4 polymer, but we have on every so often 24 to 30 sugars, we're going to have one branch point, and this branch point then has a separate bonding, and it's an alpha 1,6 between the glucose. And what this does is it allows it to have a little bit more solubility, and if I'm going to degrade, I'll have multiple ends to degrade from, whereas the amylose, or the amylose, has only one end at which we can do a degradation. The degradation of this starch is done by amylase mainly, although there is a separate enzyme that has to break the branch point itself. Glycogen, and if you take a look, I didn't even change the figure of the structure of the polymer that you see. It's an alpha 1,4 polymer that we synthesize internally. Its branch point is also alpha 1,6, but on average this is now only 10 residues on average that we have this breakage. The structures of glycogen and amylopectin are very similar in that the elongation and the branch points are the same. The only difference is how often the branch points come. And so when you take a look at amylopectin, you can see there is much less a concentration of the ends of glucose available, whereas in glycogen there is a very large number of ends for the glycogen, and in terms of degradation we will degrade from the ends, and so glycogen can be both synthesized and degraded much more quickly than amylopectin. If you take a look at the structure, the book likens it unto a koosh ball, ones that have not very many fingers and then one that has a lot of fingers. What we're really going to do is the access of glucose is linked very much to the release of the hormone of epinephrine, in which there are cases where we need glucose burst very quickly. This structural component will allow us to access glucose very, very quickly. We do not have that type of fight or flight response in plants, and so this level of branching is not needed physiologically. When we take a look at the glycogen that is in muscle, and that's what the picture is here, we also have glycogen in liver. These are the two largest stores where glycogen is seen. We can actually see this component under a light microscope, and if you would take a look, these dark nodules or granules are granules of glycogen. Very rarely do we actually see in a light microscope the things that we're talking about in biology, the actual working concentrations, but they're large. The average molecular weight is in the millions. What we find inside these granules are both the glycogen itself, but every enzyme needed to degrade and every enzyme needed to synthesize glycogen. Now, what this allows us to do is both synthesize and degrade very quickly because everything is bound, but that also requires us to regulate this very, very well so that we don't have regulation. We don't have regulating signals that will both synthesize and degrade at the same time, because if we do that, then we are in a cycle that is just using energy and gaining nothing. So we're going to find later in a chapter how we're going to have what was called dual regulation, in which one signal is going to cause either synthesis to go up and degradation to go down, or another signal that will cause the exact opposite. Now, glucose has polymers that are structural, and so if we take a look at that particular structure, we see it in terms of cellulose. Cellulose is the woody parts, the stalk, stems, trunks of plants. It is the most abundant polysaccharide in nature, the one that is synthesized most often in plants. Chitin, on the other hand, is a derivative of glucose, and the polymer there is a component of hard exoskeletons, and you find them in insects, lobsters, crabs. This makes up the second most abundant polysaccharide in the world. If you take a look at the structural component of cellulose, we just have a polymer of glucose. It is still alpha-1,4, but the linkage is beta. That beta conformation, if you take a look at the chair's formation, allows one additional thing, and that is an extra hydrogen bond between hydroxide of 1 and the incorporated oxygen of the ring. This actually gives a lot of stability to cellulose. And so in terms of, if you think of the structural components of, let's say, a tree, the cellulose making up the wood, there's a lot of bonding that holds that wood together. Now, in terms of this particular molecule, it still is a polymer of glucose, and so potentially has glucose that we could extract as energy. Unfortunately, in the mammalian system, in the human body, we do not have an enzyme called cellulase to break this down. There are ruminant animals that can break this down, and so some animals like cows can eat the grass and extract nutrition from it, but we cannot. Now, the other factor with this breakage is that there's a possibility, if you take glucose, you can refine it and you can use it for production of ethanol, as well as a foodstuff, although some people believe ethanol is a foodstuff, but ethanol can be used as a replacement for gasoline, and a country that has made that particular transition has been Brazil. And so there are active research groups going on trying to make a enzyme suitable at industrial scales and concentrations to break down cellulose. We could take grasses, we could take trees, and grasses would be renewable. And if we could harvest that and extract glucose from it, it would be a very profitable endeavor. Chitin is it's similar in structure to cellulose, the beta 1,4 bond. It has glucose. The difference is the C2 hydroxyl is converted into a amine that has an acetylated group on it. And this creates a very strong bonding. And if you think about the shell of a lobster, the entire cytoskeleton here is the structural components that provide protection to the animal. Heteropolysaccharides are classification, and this is used in a whole lot of different areas inside the body. One structural component is the cellular matrix. And so if you think about cells and the biological system, we're in water. And water is fairly fluid, but for cell integrity, what we need to do is embed the cells in a matrix that will help hold the cells together. And what we do is we get a sticky gel-like material in the extracellular spaces between cells, and what we use are heteropolysaccharides to do this. Now, if you think about thickening agents, if you've ever had your mother or grandmother or you make gravy, you can take a liquid, something like the juices that come off of a turkey when you cook it in the oven, and you take those juices and you can put them in a pan. But gravy is thick. And to thicken up the gravy, you add either cornstarch or flour, both of which are carbohydrate chains, long chains of carbohydrates. And if you put that and you mix it up, it becomes thicker and thicker and thicker. What happens is the water is attracted to all the hydroxides within the sugars, and it creates this thick gel-like material. Now, the fact that it has so much water in it between the cells with these polysaccharides, we allow the thickening to occur, but there's enough space and enough water to allow small molecules and entities to diffuse from one cell to the other, so nutrients can actually get through. The heteropolysaccharides can also form cushioning, and so the things that are between our bones to cushion the cartilage and things that soften have a large number of polysaccharides, usually in the heteropolysaccharides. Some examples of the types of things that you might see is chondroitin, keratin, and heparin. All of these also have sulfates in them. The family of extracellular matrix includes at least one N-acetyl amino sugar, and quite often there's an acid derivative. In these cases, these three, you see the sulfate, which is a derivation of sulfuric acid, and quite often we see organic acids that are attached. And so charged, hydrophobic, and they are polymers. And so you may have heard chondroitin in some commercials because of the fact that chondroitin is advertised to help people that have joint pain. The chondroitin is part of the structures that create the cushioning between bones, and so if you're lacking that, you can take chondroitin to help build back what you have worn away through arthritis or other indicators. The chondroitin will give tensile strength to cartilage, tendons, and ligaments as part of its structural component. The keratin you find in as part of the structure of the corneal cartilage, bone, hair, nails, and so this particular polysaccharide is in very structural components. And then inside the eye is a fluid, and a lot of times you get checked for the pressure in your eye. But the fluid is not just water. The vitreous humor of the eye is a very thick gel-like material, and what makes it thick is the heparin. Now, glycoconjugates, on the other hand, are sugars that are attached to either proteins or lipids. And what we're going to see is that the different classifications are going to do two different things. And so under the proteins, we got two names. You have proteoglycan, which is proteins with sugar, and glycoproteins, which is sugar protein. So it's either a protein sugar or a sugar protein. That's sometimes confusing because it seems to be saying the same thing, but this is the difference. The proteoglycans mostly have carbohydrates and some protein to link them together. So that structure is mostly sugars. And these are found on cell surfaces or in the extracellular matrix. They tend to be ones that either give that gel-like structure or give structural support. Glycoproteins, on the other hand, are mostly protein with a little bit of carbohydrate in terms of overall mass. The sugars that are attached to these proteins are highly branched and variable. And in the structure, the three-dimensional structure of these sugars that are put together create unique three-dimensional structures of which recognizing proteins bind to them. And what we're doing is placing information on the outside of cell so that can be recognized, and that allows two entities to come together and bond. It's kind of like an address. The glycolipids are similar in that they are varied sugars that are highly branched to provide information on the outside of cells. One thing about both glycoproteins and glycolipids, we find them almost exclusively on the outside of cells, never really actually oriented to the cytosol. A couple of proteoglycans, they can be bound to the membrane, so on a protein that is embedded in the membrane, keeping those sugars very close to the cell, although remember there is more carbohydrate in terms of mass or size than the protein itself. This picture given is not exactly to scale. The ones that are in the matrix to create that gel-like structure are normally secreted, and so you can see in this particular structure, we have some core branching proteins that hold on, but the majority of what's happening is all of these sugars that are branched off the protein, and this excreted into water sucks up a lot of water and makes the gel-like material. If you take a look, there are a bunch of different kinds of and types of proteins that can be in the extracellular space, and they can be attached to proteins in different ways. They can be embedded close to the membrane itself, but all of this material is creating the gel-like substance, and all of this has enough water for small molecules to be able to get through and go from cell to cell. Glycoproteins, on the other hand, are given outside the cell to give information. These particular sugars are attached through glycidic bonds, and so they're either attached to an O-glycidic bond, an oxygen that attaches to the sugar, or an N-glycidic bond, a nitrogen. There are three possibilities for attachments of sugars to hydroxides. We have three containing hydroxide-containing amino acids. We see them attached to serines and threonines. What we do not see, they are not attached to tyrosines. That's the third option that could have been possible. The N-glycidic linkages could be through several different nitrogens, asparagine, glutamine. You could go through histidine, arginine, and lysine, but what we see is that sugars are normally attached to asparagine. So what does this mean? The enzymes that are optimized to put sugars onto proteins bind to these entities, these shapes, and even though they're reactive species that are in other amino acids, the enzymes just don't bind to them to attach proteins. Sorry, attach the sugars to the protein. Now what I want you to understand about these attachments, and so we take a look up here, the attachments to asparagine, and you can see the R group of the asparagine through the nitrogen makes a glycidic bond. Here the oxygen of serine will make a glycidic bond to the sugars. What I want you to know is that in the study of carbohydrates, the shape and the color of the sugars are standardized, and so this square represents a certain type of sugar. The circles that are green represent another, the triangle represents another. And so if you study this, you could look at a chart and determine which kinds of sugars that are there, so that you know what's happening. And so we need to know the structure of which sugars are present in what order. But here's what's really highly variable, is that this sugar and this sugar can be attached in several different ways. Remember, there's 6 sugars in a glucose and 6 sugars in a glucose. We can have 1 to 1, 1 to 2, 1 to 3, 1 to 4, 1 to 5, 2 to 1, 2 to 2, 2 to 4, 3 to 1, 3 to 2, 3 to 4. And then all of those can be 1 to 1 alpha, 1 to 1 beta, 1 to 2 alpha, 1 to 2 beta. And so there are highly variable ways in which we can put these together. This is complex in terms of extracting that information, but this is very diverse in the kinds of informations that you're able to place on outside of cells, because each one of them, even though they're 2 glucoses, if they're oriented in different bonding positions, each one of those will give a different shape. Each one of those is a different piece of information. Now, what I want you to really know about this particular slide is that when sugars are placed on a protein, normally there's a core sugar that is placed, and it could be a group or a single. And so if you take a look at all of these examples, the basic T, or these 5 sugars that are placed, is the first and initial way in which sugars are placed onto proteins. And then derivatives are made from that. They can actually take off sugars and then add others, or they can just add, but each one of these represents a different informational state for the cell, which will bind to a specific binding entity. Serine and threonine have a much smaller core, which is single sugar, and then it can be derivatives can be made off of that. And so all of these are providing information. Glycolipids, we're putting a linkage of sugars, carbohydrates that are linked directly to fats, and then those fats can be embedded in a membrane. And so again, if it is embedded in a membrane, we have the cytosol here and outside the cell, where information is being broadcast by those sugars. Now, I'm talking about information. This is where we get that information translated. We have the ability to present a sugar on the outside, and then the same cell with the same sugar that is being presented on the outside can then internally bond between themselves, and that is called an intrinsic bonding. It is also called, and it's a mechanism, one of the mechanisms for intercellular adhesion, or holding together. Now, that can occur. It can be both internal, and then it can be between 2 cells, the intrinsic bonding. Between 2 cells is adhesion. That is what we think about holding 2 cells together. That's important, but probably one that is studied more often is the extrinsic type of bonding, and that's when the cell presents the sugar on the outside, and then it binds within a naturally bonding entity within the body. But this is a mechanism in which a lot of pathogens will enter in our system and then infect our cells. And so if you take a look, we have both bacteria and viruses will hijack the normal process and bind and recognize certain sugar structures to bind to cells as one of the steps in invasion of the cell to infect it. There are entities that are supposed to bind to the cell and be recognized, and so you have those types of interactions. In particular, what you're going to have is a protein that will bind to sugars. In general, a protein that binds to a sugar is called a lectin. And so the lectin will have high specificity and high binding affinity. It works like an antibody in that high specificity and tight binding, but lectins are proteins that have no structural relationship to antibodies, and they will recognize and bind to certain sugar structures. And again, here's a slide about lectins, high affinity and specificity. Again, we have both important interactions and lymphocytes and these sugars that are on the outside with a lectin, in this case p-selectin, is a way in which immunity works. The use of proteins being on the outside that bind to sugars, but bacterium, viruses, and other entities, other toxins can bind directly. Now, I'm going to show you an example of how a sugar being presented on the outside and a lectin work together in terms of molecular targeting. When looking at the animation, what we do find is the protein and sugars that are on the outside, both glycoproteins and p-selectin on the outside of T lymphocytes, are making interactions with the blood vessel. And so there's an adhesion between the two. Now, the adhesion is very similar to Velcro, in which one binds and there's another. But because of the flow of blood, the T cells don't remain fixed to one place inside the blood vessel. So what's going to happen is the volume of blood will allow this to move. It doesn't completely separate, but it breaks some bonds, and as it rolls, it'll make some bonds. And so what happens is it rolls down the vein or the artery. Now, this will happen, and it has what we would call soft or not very strong interactions if there's no signaling molecules. But at a site of, let's say, inflammation or a signal in which there's an infection, small molecules are given out that enhance the binding between the glycoproteins and the lectins such that the binding becomes tight. When the binding becomes tight, other molecules will cause, as you will see in that video, the deformation of the cell's shape and then its migration to the site that either the inflammation or the infection in which the T cell needs to work. And so here is how a glycoprotein that is on the outside of cells interact with lectins in this particular case, and how T cells work.
While red blood, these strong interactions.
An example of the information that might be displayed on the outside of cell that can be recognized is the classifications of our blood types. And so we have an AOB designation for blood types. And so, most people go into the doctor and say, what's your blood type? Well, my blood type's red. Well, that's not necessarily all that you need to know. But if you take a look, there is a lipid membrane that sugars are attached. And so we have O, we have A, and we have B. These are the three versions. If you look very carefully at the structures, O has this basic shape, A have those sugars, and B have those sugars. The difference is A has this sugar here, B has that sugar there. And so what happens is, in the body, antibodies are built to bind to foreign shapes, especially in the bloodstream. And so during fetal development, if you have B-type blood, all of the antibodies that bind to this structure are eliminated. And there's quite possibly floating around in the blood an antibody to this particular shape. And so a person that has B-type blood is fine even though it has an antibody against A-type blood because they don't have A-type blood. But if you transfuse someone with A-type blood, then the antibodies will bind to those cells and attack the blood system, and it can be deadly. The universal donor is an O antigen because the O has the basic shape of both A and B. It has the absence of two sugars, but this kind of camouflages it to any antibody, whether it's against A or B, to not recognize it. Now, how in the world do we become A type or B type, or there are people that have both A and B type? It is the differential expression of the proteins that transfer the sugars, glycosyl transferases, to that basic structure of sugar that is placed on the red blood cells. If you have neither transferase, you have O-type blood. Or though you could have a working glycosyl transferase for the A version, and then you're going to make A-type blood. You could have the gene for the B-type transferase. You can have B, or you could have both of those. And so the genetics from your parentage will guide which ones are active in you. If you take a look at all of these blood types, A, B, and O, you can see that what they're showing in the red blood cell is that the core is there but nothing else. Here they have the core plus one sugar that is different. They have both sugars presented. This has one of the sugars presented and this has the other. And it shows the type of plasma antibodies that you can have. And then you can transfuse certain things to certain people. And so for a B-type person, they can accept both O and B-type blood. For an A, you can have O and an A. For AB type, you actually have the ability to donate all of those, all the blood types to that person. And the universal donor, O, can accept O-type blood. But we can also have plasma that can be donated, and there are similarities in terms of what kinds of, that there are only certain kinds of plasma that can be donated to certain types of people. The next heteropolysaccharide that we're going to talk about is one that is structural in component. And so we're going to talk about antibodies, not antibodies, about antibiotics and the bacterial cell wall. And so there are bacteria that have this cell wall that give them an advantage. If by chance a cell comes out of the body, it is under a lot of stress, both in terms of heat and in terms of temperature and the conditions that they're under. If it's just a plasma membrane, most of the time the cells will degrade when exposed outside the body. Unfortunately, bacteria have a very strong cellular wall that allows the cell to go under all sorts of conditions and still keep integrity for the entire cell. And so if you sneeze by chance and something lands on a door handle, lots of cells die, but the bacteria has the ability to live on that, and then if you grab that handle, you can expose yourself to bacteria. And so what we're going to look at is the bacterial cell wall. If we are able to degrade that bacterial cell wall, then the bacteria can die when it's exposed to these extreme conditions. The two things that we're going to look at is lysozyme, a naturally existing enzyme in the human body that tends to react with the bacterial cell wall before it will enter into our bloodstream, and then the use of penicillin, which is a drug that was developed to combat the bacterial cell wall. And so what we're going to do is we're going to either work on breaking the sugars, which cause a lot of the stability, because the sugars are covalently linked. This will be where lysozyme works. And then the other structural component comes from a pentaglycine that is attached to a alanine, and so we have cross members that are covalently bonded in terms of protein that provide structural integrity in the opposite direction. Interestingly enough, in the Miller experiment where we take nitrogen gas or ammonia gas, CO2 gas, water, and they sparked electricity to simulate lightning, and the concentrations of the gases were in what they believed to be a atmosphere in the distant past of Earth. There was also some methane in that. Where it sparked, and at the end of the experiment, there was amino acids produced. This is the classic example for a mechanism by which amino acids could be synthesized in a natural way. And biological textbooks, especially high school textbooks, use this experiment to show that there is a way that molecules of life can be synthesized without life being around. Now, I quite often in Biochem 1 allow my students to read Miller's article originally to see what happens, but an argument is this: in the Miller experiment, there are only in the Miller experiment we get both D and L versions of amino acids, both stereochemistries. If we take a look at the human body, all we see are the L version of amino acids. We do not see any D amino acids. For a lot of creationists, the argument that L amino acids are only seen, and in mechanisms that are suggested both the racemic mixture of amino acids are made, there should logically be incorporations of both L and D in biological systems. The fact that only one was selected was a argument for the argument for design. Well, that worked for a very long time until we started looking at bacteria. And what we find is bacteria have D versions of an amino acid. And in actuality, before this reacts, before the glycine attaches, we have one more D-alanine. And so the substrate is going to be two D-alanines put together. And this is where penicillin is going to get in the way of this reaction. And so we can't use that particular argument anymore, but there are still other very good arguments for design and creation, God's design and creation. But we can't use that one argument anymore. In terms of lysozyme, we're going to take a look at two sugars, the N-acetylmuriatic acid and the N-acetylglucosamine. And so, long name, what we're going to convert these to and talk about is a sugar called NAM and a sugar called NAG. And what we're going to talk about is one is larger than the other. And so the NAM is going to be the big, and the NAG is going to be the little. And that's going to be important as we go on. Lysozyme, discovered in 1922, was actually kind of an accident. Then we now know lysozyme is found in nasal mucus and tears. Alexander Fleming was working with bacteria and unfortunately, or fortunately, sneezed during some experiments and discovered that in some of his samples the bacteria died, and didn't quite understand why. After a long discovery method, found that particularly the enzyme lysozyme in what he sneezed out was what destroyed the bacterial cell wall, which then destroyed the bacteria. Here, if we take a look at NAG, the N-acetylglucosamine, physically it is smaller, and the NAM is physically larger. And so the polymer of the sugar is a NAG followed by a NAM, followed by a NAG, followed by a NAM, and so it's a repeating pattern. The polysaccharide has a alternating sugar base, and the connection is from the NAM's 1 carbon to the NAG's 4 carbon through a beta linkage. And so it's a beta 1,4 NAM to NAG in a repeating structure. What is interesting about the specificity of lysozyme is that it will always break between the C1 of NAM and the C4 of nag. If I give it a long chain, it will only break between the C1 of NAM and the C4 of nag and make smaller pieces. And so that was a bit of information about the active site and how lysozyme worked. Now, lysozyme works on long polymers of sugar. And one thing I needed to tell you is that polynag, which is actually the structure of chitin, is actually cheap to isolate and cheap to use. Lysozyme will also break chitin. And so for a lot of experiments, we used just the polynag because it was a simple and cheap substrate. Many times when we look at experiments that are investigating enzymes, sometimes we don't use the absolute substrate, but we use a mimic. And so some of these studies that we're going to get is from the mimic itself. Now, what they wanted to do was to try to identify what was the binding pocket for lysozyme, how many sugars might have fit in there, what was going on, what amino acids were important. And so what we did is, and this is a experiment that shows, okay, let's give units of 3 nags to the lysozyme, and then 4 and 5 and 6 and so on. And we're going to look at the activity. And so remember that you only get the maximal activity once you have reached Vmax, and Vmax is reached when there's a concentration of substrate that fills all of the enzyme, creating a test tube that has all ES complex. And as soon as the product is given off, a substrate will bind, and so the equilibrium lies at 100% ES complex. And what we interestingly find is that if we feed it 3 amino acids, three sugars together, lysozyme doesn't break it at all. When we give it 4, it gets some activity. When we give it 5, it gets some activity. And when we give it 6, we're at 100%. And from 6 on, 7, 8, 9, larger, we're at 100% activity. So that means there's a break point of about 6 that represents the binding pocket as being fully saturated with the substrate. And so the 6 spots that we have 6 places for sugars to bind. Now I'll label those A through F, and if I do that and I put in the lab substrate where polynag goes in, 6 nags can fit into these spaces. Okay. Now when we break 6, the outcome was always the same. A dimer and a tetramer were formed. So there are 2 possible places in which breakage can occur. Right here to give a dimer of AB, a tetramer of C through F, or here between D and E, I could have an EF as a dimer and then A through D as the tetramer. And so this is one piece of information. How in the world do we derive which breakage occurs? Now, after some structural information was known, this is what we found out. That the site D really didn't like to bind sugars, meaning that when it bound, there was a steric interaction between the 2 sugars that is not so, that doesn't mean, or that doesn't contribute to binding. It actually prevents binding. And so, if we go all the way back to the original experiment, when I had 3 nags, I had absolutely no breakage. That means I really had no substrate, enzyme substrate binding that allowed it to be at the critical place within the active site. And so the only place that trinag would fit if there's a negative interaction at D, well, it won't be D, E, and E. It's not going to bind there because I have 2 positive energies in binding, 1 negative. If I bind here, C, D, and E, 2 positive energy values when I bind, 1 negative. If I bind B, C, and D, 2 goods, 1 bad. If trinag gets in there, the only possible place that it's going to bind is A, B, and C, because I have 3 great energetically favorable places for that sugar to bind. So 3 bind here, right in this spot. And if you go back to the original experiment, when we had 3 bind, we had 0 breakage. So what that does is eliminates B and C as a possible place in which breakage could occur. D is the place that, between D and E, is a place that actually starts to break down sugars. And if you think about that, I have a couple places in which 4 can bind. 4 can bind there, and it won't break. 4 could bind here, and it will break, because now I place something between the D and the E. Now, randomly where it binds and how it binds to it is different, and we don't see full activity when we actually give 4 binding in. Now, one other thing that we know is that structurally D is the smallest, and you have a negative interaction when sugar gets in there, but C is also small. And C is also small, but it fits NAG and cannot fit NAM. So NAG would be a positive value in terms of energetics if it binds. NAM would be a negative value. We already have D as a negative value, and so NAG is going to fit into C. Now, if I know the bond breaks between D and E, and I know NAG can only fit in C, so let's put all of this information together. Our laboratory substrate was poly-NAG. The real substrate is NAM versus NAG versus NAM versus NAG, and so I have to put the NAG in C. That places NAM in the D, and that puts NAG in the E. And if we go back to the specificity of lysozyme, it will break between a NAM and a NAG. We now know that from our experimental evidence that the place we predict breakage will occur is between the D and E site. It must orient its way in the site this way, which places the breaking part of the active site right between the place where the NAM and NAG will be broken. So this fits all of the experimental data, fits the outcome of what we see when lysozyme reacts with the bacterial cell wall. Now, how do we look at the mechanism of this reaction? Well, we kind of try to find groups that are internal to the protein, the ones on the inside, that are polar. Remember, when proteins bond, when they fold, they attempt to put the hydrophobics on the inside, the polar ones on the outside, so that it's soluble in water. Putting a polar residue on the inside of a protein is not thermodynamically stable, but we need hydrophobics in the center, and we need a few polars in the center within the active site that can actually cause the reaction to occur. Now, remember, a lot of our enzymatic mechanisms have deprotonate protonate, and so we're likely to find polar side chains that will be able to be protonated and deprotonated near the pH. But the pH can be different because inside the active site, once everything binds, it is a microenvironment, and so the pH levels and pKa's interaction can be a little bit different than what they find just floating around in the cytosol. What we find in lysozyme is an aspartic acid, 52, and a glutamic acid, 35, in the mammalian version. Remember, the numerical value of the position of these R groups may differ between species, and so the number isn't as important as knowing there's an aspartic acid and a glutamic acid. We can see that the pH dependence of this activity for this enzyme, as we vary the pH, it becomes most active around the pKa's for these particular amino acids, so that gives us an idea that is supportive that the aspartate and the glutamate are involved in this reaction. The mechanism of this, the glutamate initially reacts in such a way that it donates a proton, so it acts as an acid. And so if I didn't actually write the amino acid, I would have put H+. The aspartate, on the other hand, is not going to attack anything, but If by chance this bond right here takes this proton, taking this entire chain and making a hydroxide, what will occur is this carbon that is sp3 hybridized in the tetrahedral form is neutral. Once that bond breaks, it's a carbocation, which is now sp2 hybridized and in a trigonal planar. Now, a carbocation is formed, the aspartate that has a negative charge is very close to stabilize that. And additionally, this glutamate that was once neutral is now negative, and so we are stabilizing that particular charge of that carbocation intermediate. All right. Now, the next step, the glutamate that once acted like a acid before, in the second step, I would have written like this: acts as a base and deprotonates water. And then water will attack a carbocation. Now, if this were in an organic flask, I have trigonal planar. It's flat on both sides. Inside a organic flask, a hydroxide attack could attack from the front of the ring or could attack from the back of the ring. Now, what attacks more often is influenced by stereochemistry, and so the attack usually goes at the most the easiest stereochemistry to approach it. But since it's flat on both sides, there is no stereochemistry. There's no hindrance that will make one attack go better than the other side. So in an organic flask, the outcome here would be a mixture of both the R and the S. In terms of, if we talked about it in terms of sugars, it would be a mixture of beta and alpha formation for the attack of hydroxide. In biology, what we find is we only have one stereochemistry that is formed. The reason is we don't show it, but we understand that there's protein. And so the protein binds to this substrate and it fits in a pocket. So one side of the sugar is right up against the protein. You can't attack from that side. The other side is optimized to interact with the amino acids that cause the reaction. And so we will always attack from one side. And with a reaction that we have something substitute for another. And so before we had the sugar bound, and now we're substituting with a hydroxide. If it came in and attacked and the thing fell off, that would be an SN2 attack. The stereochemistry would be inverted. But with a SN1 attack that includes a carbocation in the mechanism, in an organic flask, it's a racemic mixture. In a biological enzymatic catalyzed, it can retain the stereochemistry that it was found in. And so we can look at the products and see and decide, is it an SN2 attack or is it an SN1 attack where I substitute, and in this case, the retention of the stereochemistry implies that we have the SN1 mechanism. And stabilized by all of this negative charge for the carbocation as the intermediate species, that is the transition state or the high energy state. And normally we talk about stabilization of charge because we have other charges in the active site to balance. There's one more reason why this particular transition state would be stabilized and allow the reaction to proceed through this mechanism. The critical elements is that glutamate that acts as a general acid in the first step and a base in the second, and then the ionized form is to stabilize the carbocation. The second part of why this mechanism goes in this particular direction is stabilization of that carbocation. Now remember, I told you the D ring actually had a negative interaction for binding. The reason is, as the sugar would come down, normally in the chair conformation, the one carbon here actually butts up and hits against the protein where there's a steric interaction between the two. Now the other 5 sugars that are binding have positive interactions and they want to fit into the active sites, but this chair in the D position hits it and gets pushed against the protein. For the sugar to fit inside that active site, what has to happen is the one carbon has to be bent up into the half chair conformation. Now, this particular structure right here is a wonderful tetrahedral, 109.5 degree angles. That's wonderful. All right. But when it's in the half chair, this becomes more parallel, okay, more like a trigonal planar. And here's the bond, one of the bonds that will be broken. Here's the other bond for the hydrogen. And so in the transition state, I have a carbocation and I have a trigonal planar structure. This is a tetrahedral forced into the transition into the shape of a trigonal planar. So if this bond picks up a proton and breaks, this hydrogen can move up, and this can be in trigonal planar, which fits much better in the active site. And so what we're doing is we're stabilizing that transition, that carbocation, by physically moving up the structure to more want to accommodate in that trigonal planar structure as being flat, so there's no steric interaction, no bad interaction anymore. So we stabilize the actual positive residue that is formed and stabilize that structural form, that 120 degrees trigonal planar structure in terms of binding. Now, penicillin is going to work in another mechanism. What we're going to do is we're going to prevent the cross-linking of the proteins that cross-link in the not parallel, but the perpendicular direction to the sugars that really create a cage of stability around around the bacteria through the cell wall. Now, there's going to be a reaction, an enzyme that will cause this glycine of the pentaglycine and this alanine to bond together. And so an enzyme is going to facilitate making that connection. And so what's going to happen is before that connection, there's going to be a leaving group that will be one more amino acid attached to the D-alanine at the end. And when the pentaglycine comes in to make the bond, the leaving group will leave, and that leaving group is an additional D-alanine. And so we're going to have two D-alanines initially right here before this pentaglycine is covalently linked. This linkage and this reaction is facilitated by glycopeptide transpeptidase. This is the enzyme in which penicillin will target to stop the linkage, creating a weaker bacterial cell wall, which allows the bacterial cell wall of the bacteria to die. Here's penicillin itself. Here's the structure. The reactive element is right here. And so this cyclohexane, this four-membered ring is very unstable. The reason is that's a tetrahedral, this is a tetrahedral, this is a tetrahedral, this one is trigonal planar. A tetrahedral wants the angles to be 109.5. The trigonal planar wants the angle to be 120. If you're in a four-membered ring, the angles are 90. There is steric strain in that bond. If by chance a reaction causes this bond to break or the ring to open up, it is irreversible. So much energy is released, it will not, the nitrogen will not react back. And so this is where the reactive species will happen. It's called a beta-lactam ring, and it's going to interact with the enzyme. And so here's the structural component of penicillin. The mechanism normally is this. The transpeptidase will react with two D-alanines. Now this is the D-alanine that will be linked to the pentaglycine. And so the first step is for the enzyme to use a hydroxide attack of the carbonyl and the D-alanine leaves. That's the leaving group. I have the D-alanine I want to attach to the pentaglycine. The enzyme will take the pentaglycine and, the pentaglycine here, and react it with the species. Electrons kick up, kick down, and the enzyme falls off, and I have my covalent linkage, a peptide bond between the pentaglycine and the D-alanine, and creates this structural stability. This is what penicillin does. Penicillin will get into the active site and react with the glycopeptide pepsidase, and that hydroxide, instead of reacting with the two D-alanines, reacts with penicillin, creating a species that will covalently link to the enzyme and deactivate that particular enzyme. If this cannot come off, it is considered a irreversible inhibitor. We've also called that a suicide inhibitor. This particular enzyme, completely dead, will not do another reaction. And so what the body would have to, or what the bacteria would have to do, is actually synthesize a new protein all the way back from the DNA, and it can't do that fast enough before the cells are, before the cell dies itself. And so that's how penicillin works. Now, why in the world would penicillin bind to that enzyme? Well, if you take a look at the structure of penicillin up here and the structure of the two D-alanines, if you take a look at this carbon and that carbon, this and this and that, this one and that one, this right here and that right there, that oxygen and that oxygen, and we circle all of this part of the penicillin and the normal reagent. Two D-alanines is the normal reagent, and penicillin. Remember, the enzyme is specific to bind to a certain entity, but what it really is, it's specific to a certain shape. Penicillin mimics the shape of the normal substrate, and so it can bind into the active site. Here, if the enzyme binds to this and reacts to it, it's reversible as part of the mechanism, but if it binds here and reacts with this, it is not reversible, and so the enzyme will die. Now, in looking at penicillin, we do have people that become resistant to antibiotics. One of the mechanisms for resistance to antibiotics is the fact that some bacteria have an additional enzyme that will break down and bind to penicillin much more quickly than the transpeptidase and break this bond. And so it doesn't fit into the active site of the enzyme, and it doesn't prevent the linkage of the bacterial cell wall, and so everything is normal in terms of its process, its life cycle, and so penicillin, that bacteria is resistant to it. A lot of times we don't actually prescribe penicillin anymore. What we do is prescribe what's called augmentin. Augmentin has an additional organic molecule that will bind to that enzyme that breaks down penicillin, inactivate it to allow penicillin time to have a high enough concentration to bind into the pepsidase and then overcome that. And so bacteria can develop proteins that will interfere with the mechanism of the antibody. How in the world do we get to that stage? A lot of times it's because of the misuse of antibiotics and the overprescription of antibiotics. Unfortunately, a lot of times treatments for antibiotics last 10 days. It's a normal course, and patients become or feel good after 3 or 4 days of antibiotics. What happens is, if they feel good, they stop taking it. The penicillin is supposed to run through the entire course so that a majority of the cells die, and as you get less and less cells, even those cells and bacteria that aren't affected by the antibiotic lose integrity because they are very dilute and they can be destroyed by that mechanism, not the action of the antibiotic, but because there isn't a colony of those bacteria that are protecting it. If by chance you get rid of a portion of the bacteria and you leave some that are antibiotic resistance, now the concentration of antibiotic resistance versus normal bacteria become larger. And so what we can do is we can challenge the bacteria and select for, allow a majority of the ones that continue to grow, only the ones that are resistant to whatever antibiotic that we have. And so unfortunately, people don't use the drugs like they're prescribed, but also people will come in to get treatment and they may have a virus, and penicillins don't work against a virus, your immunity works against that, but they have this idea that I need to take a pill to get well. And so instead of having this argument, instead of losing a patient, and instead of the patient going to another doctor to get what they want, people have in the past prescribed antibiotics as a placebo, but that again can select for things that are antibiotic resistant. We also use a lot of antibiotics in the growth of our food, and so there's a lot of antibiotics out there, and we're going to lose, and we have lost a lot of sensitivity to different antibiotics and different antibiotic classes. Trying to find out the structures of the cells, of the sugars that are on side of the cell is really kind of hard to do. And so in my graduate work, there was one person that spent their entire PhD process to identify each of these sugars and what was the alpha, beta, what the connections were, and it's laborious. Some of the techniques that we do have now are array chips. And you can use an antibody array chip in which you know the antibody that binds to a known sugar structure. And so by challenging a sample over a large number of antibodies, and you can identify whether the protein is labeled, which proteins are there, we can correlate back to which antibody was there and correlate back to what sugar was there after being released from the cell. We can do that also with grids of lectins, and these are, we can purchase those and challenge them. The one advantage is you don't need a lot of sample, and they can be reused. The deficit to this is that we have to know what the structure is to have the lectin to that to understand what is going on. But if you want to know what this structure is when you didn't have any, then we have some other types of analysis, and those are by separation, either by HPLC, and so we're using HPLC to fraction out what kind of sugars are there, and it tells you the kinds of sugars are there, but it is very hard to distinguish which kinds of bonds are they, alpha, the beta, 1 to 2, 2 to 3, and so a little bit limited, although you can isolate them and do further analysis. The other identification is mass spec, in which if you take the sugars and you separate them by absolute mass, you would know what the sugars are, what the complexes are in terms of which sugars are there, but you may not know the order because we're looking at a sheer mass, and so the sugars can be in different orders, and so it's really difficult to discover what the true structure of the sugars are. But these are signaling molecules. They interact with our immunity. They interact with bacteria. So it is important. It's just difficult to find what they are.